Hey guys, it's your friendly neighborhood TA Dakota, and I'm here to teach you guys a few things about a new technique called fluorescence microscopy. By now I know you guys have seen some pictures taken by this unique method of microscopy, but now I'm going to show you how we actually get those beautiful, vibrant colors that you guys have been seeing when you look at some of these images. Today, I'm going to explain exactly what fluorescent microscopy is, how it works, and what exactly you would do to get your specimens ready to go under the light. So what is fluorescent microscopy? Well, a fluorescent microscope is an optical microscope that uses fluorescence and phosphorescence instead of or in addition to reflection and absorption to study the properties of organic or inorganic substances. I agree, it is a mouthful. Basically, fluorescent microscopy exploits something called fluorophores. By tagging these chemical compounds onto a desired biological target, we can learn more about them. Fluorophores light up when they have UV light shined on them. They get excited, and when they come back down to a less excited state, they release photons that the microscope will pick up. The light we see emitted back is how we visualize our targeted biological specimen.
Well, how do fluorophores emit light?
Good question. It's really just a chemical and physical property that the fluorophores have. They have this ability to emit photons as they come down from their more excited state. We use that in fluorescent microscopy.
Well, how does that work?
I'm not really sure. I only have a minor in chemistry and I failed physics. But I do know how we attach the fluorophores to our desired targets, so let's talk about that. First, it's important to note that there are different methods we can use to attach our fluorophores. In regular fluorescent microscopy, for instance, there are a variety of different dyes that are attracted to specific parts of the cell. The dye stained DAPI, for instance, binds to the A to T hydrogen bonds rich in DNA. Because of this, we use it to stain nuclei in cells, because that's where a lot of DNA is.
Hey guys, it's me from the future. So I realized while I was editing this video, I completely left out a part where I was going to preface a different type of technique with fluorescent microscopy where you use a primary antibody stain. Basically, you can take an antibody targeted for a specific protein that you think is in your sample, do a wash with that primary antibody, and then there's a tag or a fluorophore on the antibody that would glow under a fluorescent microscope. So that's something we use. I show you a little bit of an example of what we're going to talk, or how that works in the video, but I wanted to insert that in now so you can kind of know what I was talking about.
Let me show you kind of what I mean by this technique. So say we have a cell, and we have a hypothesis that there's a particular protein inside the mitochondria of that cell. We don't know for sure. We do know, though, what that protein is, so we make an antibody complementary to that protein. We prep our sample, we get it all ready, and then we introduce the primary antibody. It moves all around the cell, but only binds specifically to where that target protein is. The microscope then shines a UV light all over that cell, and when it looks back, it looks to where we see a glow. Now we know that the protein we hypothesized is in fact in the mitochondria, and in real pictures we can see how much of it's there and what concentration and again what density. By doing all this, we're not just able to visualize the cell, but also specific parts and the location of some of the subcellular components.
Wait. You said the word antibody and protein, and this is an imaging technique. Isn't that just a Western blot?
You're right. Those are two parts of the Western blot process too. However, in a Western blot, you're looking to see if a target protein can be found within your sample. With immunofluorescence, you can actually visualize where that protein is and what concentration. Also, it uses a different kind of antibody staining. A primary antibody staining is used in the microscopy, where a secondary antibody staining is used in the Western blot.
Hey guys, me from the future. I was looking over the video and I wanted to clarify something else. The way I worded the primary and secondary antibody differentiation between the Western blot and immunofluorescence didn't really sit right with me. I want to make it clear that you can sometimes use a secondary antibody in both immunofluorescence and most of the time in a Western blot. What the secondary antibody does is it is actually attached to the fluorophore. So if you have a primary antibody that doesn't have a fluorophore attached, you're obviously not going to see it under fluorescence microscopy. So in that case, you would need to use a secondary antibody, attach it to the primary antibody, and then to whatever your biological target was. In a Western blot, you always have that secondary antibody, as that's the thing that comes up when you run your protein. So I want to make it clear that you can sometimes use a secondary antibody in immunofluorescence, and that's what it does. You don't just always have to.
All right, now that you have a better understanding of what fluorescent microscopy is, let's go to the lab bench, and I'll show you exactly the process of how to get your HeLa cells from the flask to fluorescence. The immunofluorescence procedure is done after you've already plated and treated your cells in the 12 well plate with cover slips. The first thing you need to do is retrieve your plate of cells from the incubator and make sure they're alive under the inverted microscope. After you've made sure your cells are healthy, you will take a 10 milliliter pipette and remove the medium from each well, discarding it in the waste beaker. Immediately after removing the medium, squirt around a milliliter of PBS solution on the sides of the well, being careful not to damage the cells by squirting directly in the middle. Just like when splitting cells, the PBS will be used throughout this procedure to wash the cells. Remove the PBS from the wells using a 1000 microliter pipette, and then repeat this process one more time, but do not remove the PBS this time until you're ready to move on to the next step. The next part of this procedure is called fixation. What we are doing in this step is adding a chemical called paraformaldehyde, or PFA, that will cross-link proteins and organelles in the cell, freezing them in place. This means as we continue the process, the cells will still look exactly as it did during this step, and thus we will know what the cells looked like due only to the treatment agents we used before this procedure. To fix our cells, we will remove the PBS and then add about half a milliliter of 2% PFA solution to each of the wells, and then close and rock the container back and forth for about 10 minutes. It's important to note PFA is very toxic, and it's important to always wear your gloves and avoid smelling or touching the PFA directly. Always dispose of PFA in its designated PFA container. After the 10 minutes is up, remove the PFA and put it in a labeled container. You will then rinse each well with PBS 4 times and discard all of this waste into the PFA container, closing the lid. The next step in the process is called permeabilization. Permeabilization means making the cell membranes more porous so that when we add our different chemicals and antibodies later, they will diffuse into the cell more quickly. To do this, we will use a detergent called Triton X100. To To permeabilize the cells, add about 0.5 milliliters of Triton into each well. Close the case and shake for 2 minutes. After the 2 minutes is up, quickly wash with PBS, making sure this process lasts no longer than 2 minutes, as not to destroy the cells completely. After you've done that, wash the wells 4 more times with PBS and discard the liquid. The next step in this process is called blocking. In blocking, we use a solution called bovine serum albumin as a nonspecific protein that passively absorbs all the remaining binding spaces on the plate. What this means is the antibodies we're going to add next won't bind to empty spaces on our cover slip, and our sensitivity will be much higher in the final pictures taken by the microscope. In this step, we'll add about 0.5 milliliter of the bovine serum to each of the wells and incubate them at room temperature in the rocker for 20 minutes. After the plate is taken off the rocker, we will now be adding the primary antibody stain. A primary antibody stain is different from the secondary antibody stain you may remember using in a Western blot. This is because the fluorophore is connected directly to the antibody in a primary stain, and that antibody will directly bind to our target protein. Our primary antibody solution is antitubulin. It will help us see the microtubules within our cells. The solution also contains trictophalidin, which will label the actin filaments in the cell red. To add the antibodies, we use a P200 pipette and remove any liquid from around the sides of each well. We are careful not to touch the cover slip of the pipette so we do not damage the cells. Before closing the lid, we will take 2 chem wipes and put them over the wells to prevent as much evaporation as we can. After this, you will then leave the plate on the benchtop for 40 minutes, checking every 10 minutes that the antibody is still on top of the cover slip. If it isn't, just suck it up with a pipette from the side of the well and put it back. After the time is up, rinse the wells 4 times with PBS just like before. is now time for the nuclear staining portion of the procedure. The nucleus of the cell is stained using a chemical called DAPI that we talked about earlier in the start of the video. DAPI is not an antibody solution, and rather a fluorophore dye that binds to areas with high AT binding in DNA. The nucleus of the cell, of course, has a lot of DNA, and thus is why we use it to stain this region. DAPI fluoresces blue under UV light and will contrast the colors from the antibody stain. Remove the last PBS wash from the step before from the wells and add about 15 microliters of DAPI solution to the wells and incubate for 5 minutes at room temperature. After the 5 minutes, add PBS to the wells with DAPI still in to dilute and stop the staining. Your cover slips are ready to be mounted. To mount your cover slips onto a slide, first put 2 10 microliter drops of molliol onto a microscope slide. Molliol is just a solution used to mount the cover slips and hold them into place on the slide. It also provides some optical properties to help the microscope later. Carefully using the forceps, you will pick up your cover slips and put them cell side down onto the dots. If there are any bubbles on your cover slip, carefully press down onto the slide with the forceps and force them out. Dry, then seal the edges with nail polish. Label the slide specifically and not the cover slips with your name, date, and what the cells were treated with prior to this procedure. Your slides are finally ready to be viewed. Go Dogs! And that's really all there is to it. What do you think, other me? Well, I thought it was interesting, and I hope you guys did too. Happy staining, good luck on your poster presentations, and I'll see you guys back in the lab soon.